All right. So, yeah, let's wait. Let's wait. You can get the cards later. All right. Couple things. So we've got a lot of stuff going on. We've got exam coming up, which I think is Thursday at 9:30 a.m. Is that right? Okay. We'll do our usual, which is get in quickly, get settled, and we will, sit in the right seats, the odd-numbered seats, as we've done before. You guys have been very good about that. So that's one thing. I am planning to do a review session, which I have not yet scheduled. I apologize for that. I will send an email out through Blackboard to the class, so you'll get an email from me announcing where. I'll also put it on the schedule page. So my tentative plan is Monday evening, but I haven't decided that. Okay, I want to give it enough time so people have plenty of time to study. Okay. All right, so I have two other announcements, both of which I've been meaning to make for some time, and I haven't gotten around to doing it. So one of which is, many of you know I'm the director of undergraduate research at OSU. I help students to connect with professors to get involved with them and do undergraduate research. I'm very passionate about that, and I'm happy to help you as well. But one of the things that I find students are not very aware of is one of the things that you can get as a result of doing undergraduate research. So I've got a big captive audience. I'm going to tell you a little bit about that today. By completing an undergraduate research project with a professor, you can get a notation on your transcript that you've done that. And that is a great value-added component to your transcript, because what it says is you're capable of not only working at a high level, but also capable of completing a project. Those things you want to get onto your transcript as soon as you can. Most people wait until they're graduating to put that on there, and by then they've already used their transcript to apply for fellowships or apply for graduate school or professional school. So my advice is get that on your transcript ASAP. When I send the email out to the class, I'm also going to send the application for that to the entire class as well. So if you haven't gotten involved in undergraduate research and you're interested in doing that, of course, I'm happy to help you. And if you have been involved in undergraduate research, the application process to get that on your transcript is very simple. If you have any questions, of course, with that, please see me. All right? So that's one thing. Second thing, again, unrelated to biochemistry, or maybe related to biochemistry, is I'm always looking for people to record my music. And I have right now a backlog of about 60 or 70 songs that have never been recorded that I'm looking for musicians who would like to record them. I pay people to do that. So the recordings that you've heard in the class have been recordings of students who've been in the class who are obviously much more talented musically than I am, and have done those. So if you are a musician and or are interested in recording for me, let me know. I can tell you a little bit more about what's involved. It's not very elaborate either. And I am looking for people to do some recordings. So if you're interested in that, please let me know. We have material to cover here, so I'm going to get through some material. I also have something for you at the end, as I always do. And since this is a time when you guys are hoping I put extra credit questions on the exam, I'm really hoping to hear you loud and clearly today. Okay? So I haven't decided whether extra credit will be on the final exam or not, so that would be useful. All right. Well, enough of the silliness. Let's get to the seriousness, which is I want to say a little bit more about vision. Okay? I was confused by my own figure last time, and so no doubt if I was, you would be too. So I want to show you this figure again. So looking at this figure, it looks like, what the hell's happened? It's got a cis bond, and it's got a cis bond over here. And when I looked at it myself in class, it was like, whoa, what is that? Well, when I went back and counted the carbons, I realized what it was showing and why it looks a little complicated. This little piece on the end, you'll notice if you look carefully, has no double bonds in it. That's actually the lysine that the vitamin A is attached to. So vitamin A is attached to a lysine within this protein. So what's happening as light hits the vitamin A is this cis bond, which is at position 7, which this definitely is a cis bond, is being converted to a trans bond at position 7. And so that's changing the shape of the vitamin A. It's also consequently slightly changing the shape of the 7-TM containing vitamin A. And that 7-TM that contains vitamin A is known as rhodopsin. Okay? So this guy here, this protein that holds this, is known as rhodopsin. Okay? All right. So sorry for the confusion on my part as well. I was a little surprised when I saw it and wasn't thinking about it. I hadn't looked at it for a while. Okay. Now, I want to say a little bit about how it is that your eye actually detects light. All right? So the change in the configuration of rhodopsin causes it to activate a G protein. Right? The G protein that it activates is called transducin. You can see it right there on the screen, transducin. And transducin is, as I said, a G protein. It behaves just like the other G proteins that we've seen. That is, it drops its beta gamma units, replaces GDP with GTP. Okay, there's actually a little GDP in there. But it's replacing it with GTP, and that activates the G protein. Now, this G protein, instead of interacting with adenylate cyclase that you've seen before, does something different. And this is usually a little bit confusing to students, so I want to try to step you through it in terms of helping you to understand what's going on. What this G protein does is it activates a phosphodiesterase. So if you think back to when we talked about phosphodiesterase last term, we talked about how phosphodiesterase broke down cyclic AMP, and that was giving you that buzz from coffee, right? Everybody remember that? Okay. So this phosphodiesterase, instead of breaking down cyclic AMP, is breaking down cyclic GMP. All right? So far, so good. That's all pretty straightforward. As it breaks down cyclic GMP, the cyclic GMP concentrations in the cell fall. Now, what I've just shown you with a light molecule is this guy can actually activate, or even multiple ones of these in the membrane, can activate thousands of these transducins within this cell. So it's possible for this to very quickly reduce the concentration of cyclic GMP in the cell. And when the cyclic GMP concentration falls, you'll see that there's a little membrane channel protein over here that holds cyclic GMP, and you can see the arrow pointing out that it's leaving. Well, it leaves because the cellular concentration of cyclic GMP is falling. All right? What does that mean? Well, when cyclic GMP leaves, this channel protein, which was open, now closes. Now, we haven't seen nerve transmission like this before. The nerve transmission we saw was opening the gates, was letting sodium in, and when sodium came flowing in, we saw a voltage change, and the nerve signal got transmitted, right? This guy starts out in the open state. Normally, cells are pumping out sodium and calcium are coming right back in. Okay? They're happily coming back in. When we close this gate, they're not going to be coming back in. Yet, sodium is continued to be pumped out of the cell. Do you understand what's happening? Well, what's happening is we create a voltage change by closing the gate, because the gate is normally open. Closing the gate causes a voltage change. Voila, we have started the signal that goes to our brain and says, hey, I just saw a photon. Now, this system is exquisitely sensitive. You can literally detect single photons with your eyes. Okay? It's a remarkable, remarkable thing. Okay? It's possible to do that. Okay? Pretty cool. Questions about that? I see a couple frowns. How would this system distinguish between frequencies, etc.? This system really doesn't. So I'm talking here about rods. Okay? So rods are really good at saying light versus no light, but they're not really good saying blue versus green. Okay? So the rods are very, very good at detecting light in low light conditions. So if you go out and you go walking across campus at night, you don't see the green leaves of the trees. Okay? Unless it's a fairly bright night. You see gray, right? And so your rods are picking that up, but you're not getting color, or at least not much color, as a consequence of that. Does that make sense?
Did you say that the system just can pick up one photon, or actually we can process it and understand?
This system can pick up one photon.
Okay, just
yeah. Yep. Yes?
Do you see what's coming out of the cyclic GMP again?
Okay, can I explain what's going on with the cyclic GMP again? I can. Right. So everybody understands how this all works up to the phosphodiesterase, right? Phosphodiesterase causes cyclic GMP to be broken down to GMP. That causes cyclic GMP concentrations in the cell to fall. That causes this channel membrane protein, which holds cyclic GMP, to lose its cyclic GMP. And when it loses its cyclic GMP, its channel closes. Okay, just a second. When the channel closes, this freely flowing calcium that was coming in, freely flowing sodium and calcium that was coming in is no longer coming in. Consequently, a voltage builds up. That voltage builds up and starts the nerve signaling. Does that help? Question back here. Is it so sensitive because vitamin A can change with a single photon? And the answer is yes, that's correct. Yep. Well, it can't constantly come out because it can only come out once, right? There's only one place for it to come out. It's not flowing through it. It's sitting there or it's not sitting there, right? Yes? No, this isn't affecting nucleotide synthesis at all. No. Yes? So this is the rhodopsin up here in green, and this is the transducin, which is the G protein. Okay? Good question. Okay. So that's how we see. It's a remarkable thing, an absolutely remarkable thing. How do we see color? Well, we see color not so much with our rods as we do with our cones. Okay? And cones have the same basic rhodopsin as do rods, with very slight differences between them. And those very slight differences between them cause them to be more sensitive to different wavelengths of light than what we saw in the rod cells. Okay? Specifically, we have cone cells that are sensitive to blue light, or more sensitive to blue light, that are more sensitive to green light, or that are more sensitive to red light. Okay? Those wavelengths of light will preferentially stimulate the process that I just gave you. So if a red cone cell picks up red light and says, hey, I just saw something here, that tells the brain red is signaling me something. Okay? The brain, of course, paints the picture that we all see. We don't see the world in red and green and blue. We see it in all colors of the spectrum. And so we can imagine all of the blending of all of those colors that's coming from all of those millions of cone cells is what paints the picture that's in front of us. Now, that's a little mind-boggling, number one. Okay? And number two, I think it starts to give you an idea about, first of all, how much information the brain is processing. Think of how many things have to go on instantaneously for your brain to paint the picture that's in front of you that's colored as we see it. Okay? And that's going on constantly when your eyes are open. Right? It's one of the reasons why our eyes use so much energy. Our eyes use a lot of energy to keep that signaling going to the brain and help us to see the world that we see. Okay. One of the things that's interesting in human beings is that in human beings, the red receptors and green receptors are very closely related to each other. Okay? The red and green are very closely related to each other. In fact, excuse me, as you can see here, the green receptor that we have actually evolved from the red one. And it evolved relatively recently, which means that they are, in fact, very, very closely related to each other. And therefore, the DNA sequence that it takes to make each one of those, all right, the DNA sequence it takes to make each one of those is also very, very similar. Okay? If we look at the receptors of chickens, for example, chickens have receptors that are different from us. You see that they're green. They have a green receptor of their own, but it didn't come from the red. it actually came from rhodopsin. That was just that generic protein that was detecting light. Okay? You see they have a violet receptor. So chickens can see other things that we can't see. All right? Mice have a green receptor, they have a blue receptor, but they don't have a red receptor. They're red-green colorblind. When you only have one of red or green, you're red-green colorblind. In fact, that's the most common type of color blindness that's there. What I want to show you now is how that happens. Okay? How do we end up being colorblind? As I said, the green evolved from the red, and the sequences themselves are very, very similar to each other. That means that it's possible for them to homologously recombine with each other, and they do. Okay? So when they homologously recombine, we can lose one of them. So if we go with this homologous recombination that occurs up here, the cell that gets this DNA is not going to have a green receptor. It's only going to have a red receptor. That person will be red-green colorblind. They can't tell red from green. Okay? Well, it turns out that this is particularly a problem for men because the proteins that code for these are on the X chromosome. And men only have one X chromosome. So it's much more likely that this recombination, if it occurs, will affect men because it only has to happen once in order for a man to be red-green colorblind. For a woman to be red-green colorblind, both sets of chromosomes have to have that recombination. And it does happen sometimes, but it's not nearly as common as it is in men. Okay. Other questions? Or questions? Yes, Eric? Is the ratio of expression of all the receptors the same? That's a very good question. You would say that if you had an awful lot more blue, for example, you might be much more sensitive to seeing blue than you were to green or red. I don't know the answer to that question. My gut feeling is it probably varies a bit between people, but I don't think it would vary tremendously because if it did vary tremendously, then you would start seeing various types of color blindness. That is, you had very few red receptors, for example. You had very few blue receptors. Then you would be very sensitive to the other two, but you wouldn't get the one that you were short on. So, but that's a very good question. I don't know the answer to that. Okay. All right. Well, we've done vision. Let's turn to hearing. Hearing is pretty cool. All right. Actually, all these are pretty cool, I think. All right. Omar? Yes. It means that you only have one red or green. Okay. So it means you can't distinguish red from green. All right? And you're just picking up one. That's all you're getting. Okay? Okay. Hearing is literally a sense that uses mechanical forces, all right, to send a signal. All right? If we look inside of a person's ears, what we see are these little tiny, what are called hair cells. And they're little bundles that are there. And it's at these bundles that the end of the nerve is. Okay? Now, if we look at this a little bit, well, there's actually a close-up of it right there. That's the bundle of these little hair cells. And what we're going to see is that as sound waves impinge upon this nerve cell, they actually change its position very slightly. Sound, of course, is a wave that moves through the air, and that wave has a mechanical force associated with it. And that mechanical force is what causes a hair cell to change its position very slightly. Okay? What we see is if we measure the amount of signal that comes out of that hair cell, as a function of how much the hair cell has moved in either one direction or the other direction, what we see is that the farther it moves, the more signal we get to a point, obviously. Okay? So it's the displacement of that hair signal, the movement of that hair, that causes the signal to happen. Let me show you this a little bit better. Okay? This shows one of those little tiny hair cells in that bundle that I showed you before. Okay? Now, notice that at the very tip of that cell, there's a little thread that connects it to the next hair cell. And if you see this hair cell, it has a little thread that connects it to the next hair cell. Everybody see that? If we look at this schematic diagram, it tells us how the movement of hair cells causes signaling to happen. If I move this hair cell, notice it's in a resting state here, and I move it slightly, I move it slightly in this direction. So let's say I pulled it to the left, okay? Then what's happened is this attachment that's between the two has opened up a nerve cell. Well, what happens when you open up a nerve cell? Ions flow, and when ions flow, you've started a signal. It's the physical movement of these hair cells that causes a signal to be propagated, or to be initiated, and then ultimately be propagated. Okay? It's the physical movement of the hair cell that causes that signal to happen. Now, if we think about this again in terms of complexity, you hear me talking. You hear a spectrum of sound. You heard me sing. You heard me whistle. You heard me talk. You heard me drop things. All of those had distinct sounds associated with them, and those sounds had great complexity associated with them. Whereas this is an on-off. This is noise, no noise. So our brain is, again, taking this pattern of signals that it gets and builds this thing that we hear as sound, as a voice, as a song, as a whatever it happens to be. And again, it takes a tremendous amount of information for the brain to process and make that thing that we hear as sound, that we understand in terms of what somebody is saying. And once again, we think about the enormous amount of information the brain is processing constantly. Without us even thinking about it, we hear. Without us even thinking about it, we see. Without us thinking about it, we smell or taste. And the last sense is touch. All those things are happening completely transparently to us, and our brain is processing all that information. It's why we say that the brain, okay, is way more than a supercomputer in terms of the information that it's handling. Because it literally is processing information much greater than any computer that we have available on Earth. That's a pretty remarkable thing. Okay. The last sense I want to talk about is touch. As I said, it's the sense about which we know the least. The perception of touch is linked to the sense of pain. And what I have on the screen is a molecule that will bring about pain. Does anybody know what this is? Hot peppers. This is the active ingredient of hot peppers. When this guy binds to receptors, particularly on your membranes, it will open them and allow calcium to flow in. Okay? It will allow calcium to flow into the cells that it binds to. They're particularly predominant on mucus-type membranes. Okay? So your tongue, your eyes, all right? Various surfaces that have that. When that happens, calcium flows in and a signal goes to your brain saying, whoa, we're in trouble. Okay? You get that sensation of hot. You may get sensation of a variety of other things that are there. There is the receptor, for what it's worth. Okay? You'll notice it's not a 7-TM, but there's a little pore. That pore gets opened up by capsaicin. It's that pore that lets in calcium that causes us to feel that sensation. Okay? Interestingly, if we take and we look at the hottest peppers on the face of the earth, okay, and there are some really, how many people like really hot stuff? Yeah, okay. I had a student that wanted to get, what's it, the ghost pepper? Okay. So I've actually seen him eat a hamburger with ghost pepper on it. I mean, he was even he got a little, whoa, that was much, okay? But he was a remarkable young man, okay? I didn't think that was, I thought you would almost feel it just by sitting across from him. You know, you breathe the fumes of this thing and you get it. What's interesting is that mammals are all pretty much susceptible to that. If you give it to a dog, that would be not a good thing to do to a dog, let's put it that way, okay? But birds are not. Birds are completely unaffected by it, okay? And it turns out that for peppers and various plants that have this compound, all right, their dispersal is frequently by birds. They eat the seed, it passes through their system, they poop it out, and a new plant arises somewhere else. It's a really good case of sort of co-evolution of two things happening, where this compound is affecting everybody else who might not go and help spread this seed in its best possible way. But birds have evolved an ability to do that and also help the plant in the process, so it's kind of cool. Okay, that's actually all I have to say about touch. So there's really not much more I can tell you about touch. But I will stop and take questions. Yes?
So if he had a really high tolerance for the capsaicin,
Yes?
does he also have a high tolerance for pain?
Okay, if you have a high tolerance, it's a very good question. She just asked if you have a high tolerance for capsaicin, do you have a higher tolerance for pain? I can't answer that question directly because I don't know the answer to that, but I will tell you something related to it. And the relation to it is that people who do pain work now are actually using capsaicin as a way of relieving pain. And I don't know fully how it's done. I believe that they actually inject it into the bloodstream. I'm not sure of that. And what they're doing is that you're not so consciously becoming aware of it, but your brain actually is. And so it's sending a signal to the brain that the brain starts to ignore. And because it starts to ignore that signal, it ignores other pain signals, and you don't feel as much pain associated with that. So that's related to the question that you've asked, but I don't know if a person who has that has a lower or a higher pain threshold. I just don't know. Yes? Yes?
I'm kind of confused on the brain's ability to distinguish between two sounds or how one sound will remain consistent. Because if it's the only way that we can detect sounds is by the movement or opening of
Yes?
hairs, then that could be variable from one moment to the next, couldn't it?
It is variable from one moment to the next because every word I'm saying is variable. It's a continual change.
So what results in the consistency of your voice? Like I can hear your voice the same today as I did.
Okay, so it's kind of a complicated question. So his question is, well, how is it that I don't hear this sound in discrete units? I think that's basically what you're saying. And the answer is because you have millions of these cells. All right? So your brain is much like it's painting the picture that you see as vision. It's also doing the same thing with sound. So it's picking up continually different signals from different ones and giving you that sort of continuity or the appearance of continuity of what you see or what you hear in this case.
So each frequency has like a fingerprint of how it affects the hairs?
Very good question. So each audio frequency has a fingerprint associated with it, and the answer is yes, it does. And that's how the brain actually does it. It's remarkable. Yes, Tyler.
So on vision, it's on the X chromosome. This is hypothetical. If you're a woman and you have one X chromosome with the recessive color blindness, but the other one does not, is it necessarily a dominant trait, or do you get an intermixing of slight color blindness?
Okay, so this question really relates to the degrees of color blindness. Would a woman who had one red-green and one red, let's say, experience color blindness? She probably would be a little bit more sensitive to that because, again, it's a numbers thing, like Eric asked earlier, that you would probably be making more of one receptor than the other, and that would influence the amount of color that you would see. Yes?
Another question on vision. So when you stare at the sunlight and then you close your eyes,
Oh, boy.
you still see it. Why is that? There's no more photons.
Okay, so the question is, if you stare at the sunlight and you close your eyes, you still see the sun. I can relate to this as well. I like to jog, as you know, and on rainy days I jog on a treadmill we have at home. And the treadmill has a very bright green screen. And when I'm done with that treadmill, I look up and I can see this pink rectangle, for about a minute or two after that. The answer is that the cells are fatigued. So you've overstimulated the cells, and they're still signaling to some extent. That's basically what's happening with them. Yep. Okay. Yes, back in the back. You have a sound that you don't like to hear? You mean like my voice or what? Okay, so desire, liking, or something is all a function of our brain. Okay? So our brain is ultimately telling us there's something wrong with that. The same way that our brain is telling us you've eaten something that's poisonous, vomit. Or something that's not good for you, vomit. That's why those sorts of things are there. So that's actually a function of the brain, not of the senses themselves. Yeah? Do rods not have cones? Well, if you look at the, back to the, I'm sorry? Yeah, okay, but I'm going back here to this. Okay, so most animals have a setup like this, where they don't have the ability to get red. Okay, so dogs would fit in this category as well. Yeah.
So corrida with red is of course doesn't really, it can be any other color, right?
Well, so the question is, would they see it as, I mean, so they're not distinguishing that color from, let's say, a green of the same thing. Yeah. That's all it means. They still see it. They just don't see it as we see it as a distinct color. Yeah, that's what color blindness means. Yes? Taste?
Yeah. So you know how we told 65 things are animals?
Oh, that's a good question. Do animals only have the ability to have 5 senses of taste? My suspicion is that they would, but I'm not a veterinarian, so I couldn't tell you that. But I would be surprised if it were significantly different from our own. They can certainly smell differently, though. Well, we've reached a point where I should go talk about the immune system now, right? So one of the surprises I always have for the class is, usually I get to the immune system because that's where I'm at. But this year we had a day off, and so I'm not even going to get there and take you through the torture of running through the immune system at breakneck pace, and at the very end saying, oh, by the way, we're not going to cover the immune system, which is what I usually do. And it makes me very popular, very unpopular for about 20 minutes, and then very popular at the end of that. But rather than do that today, I think I won't bother to try to do that. The other question I always get is on the eCampus, after students watch this, they say, is this true for the eCampus too? So I will officially say, eCampus, you don't have to do the immune system either. So that way I won't get 50 emails from the eCampus asking me, we have to do immune system. So we are at the end, and you guys have been a very, very good class in several ways. I've had more questions in this class than I've ever had, as I've said before. I'm just delighted with the interest of all of you in this subject. I apologize that I'm not an expert in many of the areas that you've asked, but where I can, I'm always happy to try to answer, and I love the curiosity. It's really been wonderful. You guys have been a very, very good class. I've really enjoyed that. I've got some songs for us to sing at the end, and I had something cool yesterday. I got a brand new recording of one of my songs. It's not a biochem song, but it's a beautiful recording. And I'm going to save that for the last thing that we're going to do today. So I've got 2 songs before then, and then a recording at the end. But before that, I have a young man named Tyler who has a public service announcement of some sort that's in the form of a song as well. So I'll ask you to give Tyler your attention, then we'll turn to the metabolic melodies.
Hey everybody. So what I'm going to be doing, my public service announcement, during the first 3 days of finals week with the possibility of extension, I'm going to be having a booth outside the library. It's basically just a relaxation booth for you guys. I know finals week is tough. This test isn't going to be tough because we love the material, obviously. But I'm going to have pillows so you can sleep or fight. That's up to you. But there's candy. There's going to be some CDs with jokes and songs on them that I've made myself. Everything's going to be free. Donations, of course, are accepted but not required. If you want a T-shirt, such as this is a prototype. It says prototype funny T-shirt, insert image here, but I can make one on the spot for you just if that makes your day. It's just to cheer you up for final. So to get that built up, I wrote a song about biochem, and this is dedicated to the eCampus students and to what I like to consider myself a pseudo eCampus student. So here we go. I am a bio major, and so I understand. The knowledge in this class is required, but still my time is in demand. Wait, what's that you say? YouTube is here to save the day. I've seen so... Can I say goddamn?
No.
I've seen so goddamn many online classes. I may as well be a member of eCampus. Wait, what's that you say? Ahern heard you skip, now he's docking your grave. Wait, I didn't write that last line.
No. Okay. I think I'm going to keep this and sell it on eBay or something, I think. I don't know. But thank you, Tyler. Whoa, I just lost my microphone. Okay, now for the fun part. This is where I want to hear you guys sing loud, right? We're going to start with a song about vision, all right? It's to a tune you all should know. It's called the Vision Thing. It's to the tune of the Star Spangled Banner. Did you know you can see in the dimmest of light with your rods and your cones and their retinaldehyde found in rhodopsin? It's got a bond shaped as cis, but it changes its state when a photon gets it straight. Then the signaling kicks in, thanks to a transducin, 'cause its GTP ways turns on diesterase. So gated ion channels stop charges from passing through, such as sodium +1 and calcium +2. Awesome. All right. Okay, the next one is a long song. It's a very easy one to sing. You may not know the tune originally. It's to an old tune called Those Were the Days, My Friend. Anybody know that song? Those were the days, my friend, we thought they'd never end. We'd sing and dance forever and a day. Right. Old Russian song, actually. Okay, so this one is called Central Dogma Zen. It's a long song, so we gotta stick with this one. Once upon a time, a cell decided the time was ripe for it to split in two. Had to copy cellular instructions, for the daughter cell would need them too. Bring in a helicase, unzip the DNAs to ease the stress. A gyrase joins the fray. Strands must be held apart. SSBs do their part, and primase builds a primer RNA. Sliding clamp comes in behind clamp loader. DNTP's floating all around. In the cells, a replicase is waiting for the chance to start another round. Polymerase, my friend, starts at the 3' end. It puts a T across from every A, a G across from C. Perfect simplicity. The leading strand is made in just this way. The lagging strand is made in little pieces, Okazaki fragments you recall. Pol 1 fills the gaps that lie between them. Ligase comes in next and joins them all. Blueprints can't have mistakes. That's why polymerase corrects its work with exonuclease. Proofreading one by one till all its work is done. Hooray for DNA polymerase. An organism's cellular construction, with blueprints for the things they have to do, requires converting DNA instructions to ribopolymers. Oh yes, it's true. Because they've been bestowed with a genetic code, the RNAs provide the cell with means to link amino A's in most directed ways inside the protein-making cell machines. If coli cells don't have galactosidase, and lactose should appear inside its food, the lac repressor leaves the operator, 'cause otherwise metabolism's screwed. Polymerase unwinds the DNAs it binds adjacent to the start site where it docks, unravels A's and T's with such amazing ease at the promoter's little ta-ta box. The process moves along without much trouble while making RNA inside the cell. It all occurs inside transcription bubbles, where bases get linked anti-parallel. mRNA then roams to find the ribosome, subunits large and small by near the end. The AUG's in place inside the P site space. Initiation you can comprehend. The mechanism shifts to elongation, proceeding by 3 bases at a stretch. A GTP is required for translocation, advancing 5 to 3, the whole complex. The process moves anon until a stop codon arrives and causes movement to suspend. Translation has to cease, a peptide gets released, and we have reached the central dogma's end. All right. Thank you, guys. That was good. That was very good. Okay, the last one, probably not going to sing. I'll put it up if you want to sing it. I've actually got a recording. I'm very pleased with this recording that I was sent yesterday by a former student of mine, and hope you enjoy it. It's all about tweeting. It's called Tweet. And where are we going here? Get it started. It's an old song. All right, guys, that's the end.